Hello everyone. Today I'll talk about flow cytometry. So, what is flow cytometry? It is a technique which is used for counting, examining, and sorting microscopic objects which are suspended in a fluid based on their optical properties. So, what it does is it helps in simultaneous measurement and analysis of multiple physical characteristics of single object as they are flowing in a fluid through a beam of light. It consists of three systems: fluidics, optics, and electronics. Okay, let's take a look at this picture where we can see this is a picture of flow cytometry. So the sample is introduced and it comprises of stained cells in suspension. Okay, this is the area where the sheath fluid is entering inside. Okay, this is the nozzle. And then because of the shape of this structure, what happens is there is hydrodynamic focusing and cells pass through in a single line. So as they pass through it, what happens is we are giving a laser light source, and this laser light will pass through the different particles present in this hydrodynamic focusing flow. So some of the light will be scattered, whereas scattered what? Laterally. Some will go directly straight. So fluorescence will be emitted from cells and it will be detected. And there will be forward and side scattered light from all the cells which are detected from this sample. So what happens in a flow cytometer? Number one, fluidics. The cells in suspension flow in a single file through an illuminated volume where they scatter light and emit fluorescence. Second is optics, that is collected, filtered, and converted. Okay, it will collect, filter, and convert the uh, light. And then electronics will interpret the results. It will convert it to digital values that will be stored in a computer. So again, fluidics, it will help in transport of cells in a stream to a laser beam. For what? For interrogation. And optics, through optics, the lasers will illuminate the cells in the sample stream, and the optical filter will divert light signals to the appropriate detectors. So finally, it will come to the electronics part where it will be converted to detected light energy, light signals into electrical signals. Okay, there are two scatters, the forward scatter and the side scatter. What are their roles? The forward scatter is involved in it is the amount of light which is scattered in the forward direction, and it helps as an indicator of relative the size of the cells. We can tell the size of the cells from the forward scatter, whereas the light scattered at right angles will be side scatter, and it gives information about the internal complexity and the granularity of the cells. So if the cells are more complex and more granular, there will be more side scattering. This is a very important picture. As we can see, the sample is entering into the flow cytometer. Through the sheath fluid, it is going through the structure, and then hydrodynamic focusing is helping in making it a single line of cells. And then these cells, mm, the laser beam is focusing over the cells and the rays are getting scattered. Okay, so finally, this is the optics part where mirror filters are there from where the light is, the fluorescent light is detected, and then various graphs are made. Okay, this is fluorescence, different color light is formed. And then next come to B, C, the laser is going through the cell. This is the cell is there. Okay, this is the cell with its cell organelles. So some of the light is going straight away. That is the forward scatter. Okay, and this forward scatter will help in knowing the number of cells. Hmm? And if it is going through the different complexities or the granularity, then there will be side scatter. Okay, and then it is detected. The side scatter is detected by another detector and the forward by the other detector. And then data acquisition and analysis in the computer software. This type of graph is formed because of different color of fluorescence, right? And see, it's a very good example where granulocytes, which are more granular, so obviously side scattering is more here. But lymphocytes, they are not, they are agranulocytes, right? So lymphocytes, they have a lesser side scatter. So this is a very nice example given here. So what is fluorescence? When a fluorescent compound absorbs light energy over a range of wavelength which is characteristic for that compound, it causes electron to raise to a higher level. So when it goes to the higher level, there is the excited electron, it quickly tries to come to the normal state, to the ground state. And when it does, there is release of energy, and this energy is in the form of photon of light. So this is nothing but fluorescence. Okay. And lastly, what are the applications? Where is the matlab, flow cytometry is used? One is cell sorting, second is DNA quantification, third is analysis of cell size, fourth is cell cycle analysis, and lastly, immunophenotyping. So that's all for today. See you in next class. Thank you.